Welcome back friends. Welcome back in another video tutorial for microbiology with Shakni. So today in this video tutorial we will going to talk about different catalytic strategies that enzyme employ to complete their catalytic reaction. So here we will going to talk about three important catalytic strategies. First is covalent catalysis. Then we will going to talk about acid base catalysis. Then we will going to talk about metal ion catalysis. And at last we will going to talk about two important definitions related to catalytic strategies of enzyme. So let's get started with the video. So enzyme can employ one or more than one strategies among the three that we have discussed. So for example, if we talk about chymotrypsin, it uses both covalent catalysis and general acid base catalysis. Although we have already discussed how chymotrypsin works in our enzyme mechanism of action video, so I hope it will be now easy for you to understand what is covalent catalysis and general acid base catalysis as well as metal ion catalysis. So that is the idea. So these are the common type of catalytic strategies that enzyme employs. For example, general acid base catalysis, metal ion and covalent. And that does not mean that a particular enzyme will going to use any one of them. It may use any two of them or all the three. Anything can be happen, or it may also use any one of them. Okay, so first let's talk about the discussion on covalent catalysis. So in covalent catalysis, remember, in between the active site of the enzyme and the substrate there will be formation of reversible covalent bond. Okay, that means the substrate and the active site interaction will be reversible covalent interaction. And remember, this type of covalent interactions are temporary, but substrate will be bound to the enzyme during the course of catalysis. Okay, so if we're going to talk about covalent catalysis in details, then first of all, we have to learn that for performing covalent catalysis, enzyme should have a important thing in its active site. That is, the active site of the enzyme should contain either nucleophilic center or electrophilic center. Okay, and depending upon this, whether the enzyme contain nucleophilic center or electrophilic center, we can classify covalent catalysis into two class, sorry, here we have to say two subclass. So these two subclass are covalent nucleophilic catalysis, another one is covalent electrophilic catalysis, right? So in case of covalent nucleophilic catalysis, remember, the active site of the enzyme should contain negatively charged nucleophilic group and it will going to attack electrophilic center on the substrate. Okay, and in case of covalent electrophilic catalysis, the enzyme will contain electrophilic center and it will going to attack the nucleophiles on the substrate. Okay, so that is the idea. So here I have written all the things that I have explained to you. So that is the thing. So you may take a screenshot for making for further use of this. Okay, so now here, for covalent nucleophilic catalysis, the enzyme should contain negatively charged groups or nucleophiles, right? So they include negatively charged oxygen, sulfhydryl groups, carbon ion groups, imidazole groups, etc. etc. Okay, if enzyme contain this type of negatively charged group, then this negatively charged group will going to readily attack the substrate having electrophilic centers. Okay, and those electrophilic centers are phosphoryl groups, glycosyl groups, acyl groups. So that is the idea of covalent catalysis. So in covalent catalysis, the enzyme will contain negatively charged group and it will going to attack the electrophilic center of the substrate, right? And here we have to remember the examples of what kind of electrophilic center should be present in the substrate and what kind of nucleophile should be present in the enzyme. That is the idea. Okay. Now, in case of covalent electrophilic catalysis, the case will be completely opposite, right? Here, electrophilic center should be present on the active site of the enzyme. For example, in covalent electrophilic catalysis, these electrophilic centers will be formed by coenzyme adduct. Okay, that means here a coenzyme should be required, right? That will generate the electrophilic centers in the enzyme. But in case of covalent nucleophilic catalysis, it is not that much required. Okay, so that is the idea of our covalent catalysis. So here we have to remember just two things. One is we are classifying covalent catalysis in two classes. One is covalent nucleophilic catalysis, depending on the nucleophiles or negative charge on the active side of the enzyme. For example, sulfhydryl groups, oxygen group, carbon ion group, imidazole group, that will going to readily attack on the electrophiles on the substrate, right? And another one is covalent electrophilic catalysis, where coenzyme will going to generate a electrophilic center on the enzyme, right? So that is the covalent catalysis. Now let's move on acid base catalysis. So first thing in acid base catalysis, remember here transfer of proton is involved. Okay, and here acid base catalysis are of two types. One is specific acid base catalysis, another one is general acid base catalysis. Remember, in specific acid base catalysis, hydronium ion and hydroxide ion act as a acid and base. And in general acid base catalysis, except hydronium and hydroxide ion, other things will going to act as a acid and base. And these other things are mostly the functional groups present on the amino acid side chains. Okay, because enzymes are nothing but proteins, right? And proteins are nothing but amino acids, and there are variety of amino acids present that contain charged side chains, right? So those charged side chains will going to act as a acid and base. So that is the idea. So here, as you can see, acid base catalysis involve proton transfer, right? So specific and general acid base catalysis are the two types. In specific acid base catalysis, hydronium ion and hydroxide ion act as a acid and base group. And in general acid base catalysis, remember ionizable functional group of amino acid side chain and of cofactor. If the cofactors are present, then they will going to act as a acid and base. Functional groups such as imidazole group, we know that in histidine imidazole group is present. Hydroxyl group, you may find hydroxyl group in threonine. Carboxyl group, for example, our glutamic acid, aspartic acid. Sulfhydryl group, for example, cysteine. Amino group, for example, lysine. Phenolic group, for example, your phenylalanine, etc., etc. In those type of amino acids, their side chain may act as a acid and base in acid base catalysis. Okay, so that is the idea. And now remember one thing, that a enzyme generally performs general acid base catalysis. Okay, specific acid base catalysis is usually not observed in case of enzyme. So an example of acid base catalysis is the hydrolysis of peptidoglycan by lysozyme. Okay, we have also discussed the mechanism of lysozyme, right? So you may also watch that video to understand it. And another important thing, in case of specific acid base catalysis, it depends on only on pH. Here buffer concentration do not create any kind of effect. But in general acid base catalysis, it depends on pH as well as buffer condition. Because remember, buffer here may act as a acid and base in the transition state, and thus it can affect the rate. That is why in general acid base catalysis it depends on buffer concentration, but in specific acid base catalysis it do not depends on buffer concentration. So that is the idea of our acid base catalysis. So acid base catalysis, in acid base catalysis we have to talk about two things. One is general acid base catalysis, another one is specific acid base catalysis. In general acid base catalysis, remember, except hydronium and hydroxide ion, all other things may act as a acid and base, but in specific acid base catalysis, only hydronium and hydroxide ion act as a acid and base. In general acid base catalysis, remember, the charged amino acid present on the enzyme will going to play the role of acid and base, especially their functional group. Okay. And in general acid base catalysis, remember it depends on pH and buffer concentration. But in specific acid base catalysis, it only depends on pH. So that is the idea of our acid base catalysis. Now let's talk about metal ion catalysis. Metal ion catalysis it is also known as Lewis acid base catalysis. So we all know that many metal ions are present that are very important for many enzymes, right? And on the basis of this, we can classify enzyme into two parts. Okay, one is metalloenzyme, another one is metallo activated enzyme or metal activated enzyme. Okay, so metalloenzyme are those where the metals are bind to the enzyme very tightly, and their metal ion is necessary for the enzyme to maintain its stable native state. Okay, that means in case of metalloenzyme, remember metal ions are their very important part, not only for catalysis as well as for maintaining the structure of the enzyme, right? But in metal activated enzyme, remember that metal ion is only important for catalysis, not for maintaining the structure. Okay, now this metal ion have various type of function during any kind of enzymatic catalysis. For example, this metal ion act as a electrophilic catalysis. It act as a electrophilic catalyst, and it also stabilizes the negative charge or ne- any it stabilizes the negative charge or electron density on a reaction intermediate that formed during reaction. We have seen this type of things during our discussion on mechanism of action of enzyme discussion, right? So another potential function of metal ion is they provides powerful nucleophiles at neutral pH. So they act as electrophilic catalysis. They stabilizes negative charge or electron density on a reaction intermediate. Along with that, they also provide powerful nucleophile at neutral pH. So that is what we have to remember from metal ion catalysis. Okay. So that is the three type of catalytic strategy that an enzyme may employ for doing its catalytic job, right? It may employ general acid base catalysis, it may employ covalent catalysis, it may employ metal ion catalysis. Okay, so remember again very carefully, an enzyme may employ more than one of the three catalytic strategies or may employ only one. Okay, that is completely depending upon that particular enzyme. Now, two important definition that are very important from enzymology. So first one is enzyme unit and another one is cattle, or what do you mean by cat? So enzyme unit, remember both the definition are for specific enzyme specificity, okay, or measurement of enzyme, because enzymes are present in very small amount. That is why we used to measure them on the basis of their activity, okay, rather than measuring them just only on the basis of gram, milligram, etc. Okay, for example, if you see the definition of enzyme unit, here we are saying that the amount of the enzyme causing transformation of 1 micromole of substrate per minute at 25 degrees centigrade under optimal condition of the measurement, that is enzyme unit. And in cattle, it is the amount of the enzyme causing transformation of 1 mole of substrate per second, that is cattle. Okay, so that is the idea of enzyme unit and cattle. You can see here we are measuring the enzyme on the basis of their activity. Okay, so that is very important. So that is our today's video. So here if we going to do a quick recap, so here today we have discussed about the three catalytic strategies that an enzyme employ to do its catalytic job. Okay, it may employ any one of the three or it may employ more than one, right? So these three catalytic strategies are acid base catalysis, covalent catalysis, and your metal ion catalysis. In covalent catalysis, we have seen that covalent catalysis can be divided into two subclass. on the basis of whether the enzyme contain electrophilic center or nucleophilic center. Remember, if the enzyme contain nucleophilic center, then for doing covalent catalysis, the substrate should have electrophilic center. Similarly, if the enzyme contain electrophilic center, then for doing covalent catalysis, the substrate should have nucleophilic center. Okay, so that is the idea of covalent catalysis. Then we have learned about the acid-base catalysis. There we have learned two type of acid-base catalysis. One is general acid-base catalysis, another one is specific acid-base catalysis. In specific acid-base catalysis, hydronium ion and hydroxide ion act as a acid and base. And in general acid-base catalysis, remember it very carefully that except hydronium and hydroxide ion, other things, mainly the functional group of the amino acid, act as a acid and base. Okay. And in metal ion catalysis, remember metal ion will going to play a very important role in catalysis. For example, it may act as a electrophilic catalyst or it may act as a nucleophile, okay, at neutral pH. Okay, so that is the idea of our metal ion catalysis. And then we have discussed about the two definition, that is the measurement of the enzyme on the basis of its activity. One is enzyme unit, another one is catalyst. So that's it today's video. So I hope this video will be very helpful for you. So thank you for listening to this class.